Hello, everyone, and welcome to today's presentation, Using Human Stem Cells to Model Neocortical Gyration Phenotypes, presented by Ashleigh Schaffer, Assistant Professor in the Department of Genetics and Genome Sciences at Case Western Reserve University. I'm Kate Judd of Thermo Fisher Scientific, and I will be your moderator for today's presentation. Before we begin, I would like to remind everyone that this event is interactive, so we encourage you to participate by submitting any questions you have at any time during the presentation. All you have to do is click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please also use the Ask a Question box to let us know. Now, please allow me to introduce Ashleigh Schaffer. Ashleigh graduated with a BS in genetics from University of Wisconsin-Madison and went on to earn her Ph.D. from the University of California, Irvine, under the mentorship of Dr. Maike Sander. She continued her training as a postdoctoral fellow at the University of California, San Diego and Howard Hughes Medical Institute under the guidance of Dr. Joseph G. Gleeson, where she focused on elucidating the genetic cause and molecular mechanisms underlying recessive pediatric neurological disease. During her time with Dr. Gleeson, Ashleigh discovered over 20 novel genetic causes of disease, many for recessive, syndromic pediatric neurodegenerative disorders. She joined the Department of Genetics and Genomic Sciences at Case Western Reserve University as an assistant professor in February of last year. I will now turn the presentation over to her. Ashleigh?
Thank you. I'd like to start by thanking Thermo Fisher and the organizers of the event for giving me the opportunity to talk about our use of human stem cells to understand neurological disease mechanisms. I'd like to start by drawing attention to these different brains on this slide. You can see that Drosophila, mouse, and human brain have very distinct key features that result from differences in development. The most obvious difference is the size of the brains, while another is the presence of cortical folding or gyration pattern. This feature develops from a radial unit hypothesis, so neural progenitor cells proliferate and give rise to intermediate progenitor cells, which can transiently amplify and differentiate into neurons that migrate out of the cortical plate to establish and integrate circuits. You might notice from this review that humans have an additional cell type not commonly found within the mouse brain, called the outer radial glial cells. These cells can also proliferate, greatly expanding the number of neurons that can develop from a single ventricular progenitor cell division. Due to these differences in brain development, certain human disorders are not well modeled in mice. Here I'm showing an image of a coronal adult mouse brain section. The mouse on the left is wild type, and the mouse on the right has a homozygous mutation in CKD5RAP2, which is a known causative gene for primary microcephaly. The bottom panels are a zoomed-in image showing very little phenotypic difference between mice with and without this human disease-causing gene mutation. In contrast, in the panel on the right, Madeline Lancaster from the Noblique lab developed and induced pluripotent stem cells from patients with primary microcephaly that have mutations in this very same gene. When she differentiated them to cerebral organoids, or mini brains, she saw a clear phenotype in the dish. The patient mutation impaired proliferation of neuronal progenitor cells and led to precocious differentiation. This result suggests that animal models might not be a suitable choice to study all types of human disease, and that using stem cells may be a valid alternative approach. So to use stem cells as a disease model, there are typically two approaches that we use. The first is to develop induced pluripotent stem cells from patients that have a specific disorder. This method is particularly useful when the genetic cause of the disorder is not yet understood, or for complex genetic disorders with many contributing variants. The disadvantages include differences in the genetic background between the samples tend to lead to large experimental variation. The second approach is to use human embryonic stem cell lines and use a genome editing technology like CRISPR-Cas9 to introduce disease-causing variants. This method is useful when the genetic cause of the disorder is defined and only consists of a few mutations or of a specific haplotype. By introducing these mutations into the stem cells using gene editing technology, we can create isogenic stem cell lines. In this case, the genetic background of the experiment is tightly controlled, and we expect to see less experimental variation. We can then use these stem cells to develop neuronal cells using well-established differentiation protocols. We can make organoids, and we can also make pure populations of neurons that can be used for biochemical analysis. So now I'm going to tell you about a story where we applied stem cell modeling to understand a neurological disorder that could not be well modeled in mice, lissencephaly. It's a developmental disorder that's caused by defects in neural migration. In this simplified coronal image of a human brain, affected individuals present with a smoothening of the cerebral cortex upon MRI imaging. This is referred to as pachygyria or agyria. Most patients also have mental retardation and early onset of intractable seizures. Lissencephaly is a very rare condition, but it's usually genetic, with roughly 90% of all patients having mutations in one of the three common lissencephaly-causing genes, LIS1, tubulin, or DCX, which I'll talk more about in a couple of slides. So lissencephaly is not well modeled in mice. Deletion of known lissencephaly-causing genes, like DCX, does not produce a cortical phenotype. Initially, it was hypothesized that mice might have added redundancy, so researchers decided to use shRNAs to try to knock down multiple isoforms to study migration. Unfortunately, we now know that shRNAs can also induce neuronal migration defects on their own by dysregulating the microRNA processing pathway in neurons. So as I mentioned in the introduction, neurons are produced at the apical surface of the brain through asymmetric cell division of neural progenitor cells or glial cells. The fated neurons migrate in an outward fashion, and they populate each layer of the cortex in a temporal manner to produce a 6-layer cortex. When you have aberrant migration, you end up with a smooth, thickened 4-layer cortex, and patients with lissencephaly have this feature. So neuronal migration has been studied for decades in animal models, but only recently have we been able to link human mutations to a lissencephaly phenotype. In this structure of a basic bipolar translocating neuron, we can identify various mutations that affect a variety of processes. For instance, mutations in relin, VLDLR, and ApoER2 tend to affect growth cone control by altering recognition of guidance cues. Other genes like CDK5 and P35 alter cell signaling cascades that affect multiple cellular processes. Recent hypomorphic mutations in the tubulin proteins, which are the structural proteins that make up microtubules, have been shown to cause a syndromic patient phenotypes with pachygyria. The first gene mutations in patients with lissencephaly were in the genes LIS1, or a deletion of the tip of chromosome 17, which causes a disorder called Miller-Dieker syndrome, as well as the DCX gene, which causes lissencephaly and subband cortical heterotopia. This class of proteins in the gray box controls microtubule dynamics and function in nuclear translocation during migration. So as you can see from this image taken from a review by Bill Dobbins, each gene and mutation type creates a unique form of lissencephaly that can be clinically categorized by anterior to posterior gradient, as well as the severity of cortical smoothening. I want to note that you can sometimes guess what the affected gene will be based on patient MRIs, because DCX mutations always cause an anterior to posterior gradient, and LIS1 or Miller-Dieker syndrome deletions always cause a posterior to anterior gradient. When we look at patient MRIs and see unique patterns, we can be fairly certain that a novel causative gene underlies the patient disorder. So to identify new causative genes for pediatric cortical phenotypes, we start our research pipeline by recruiting patients with interesting phenotypes, like unique MRI findings in this case. We can then perform exome or genome sequencing to identify new causative genes or find candidate variants that might underlie the disorder. We then perform functional assays in the lab to assess the pathogenicity of the variant and determine the mechanism of disease. We will take this information from the lab and work with our collaborating physicians to reassess patient treatment options, improve disease classification, and better define clinical syndromes. Using this approach, we identified 3 families with a unique MRI showing a diffuse pattern of pachygyria with cortical thickening up to 10 times its normal thickness. Patients had a thin corpus callosum, varying degrees of cerebellar hypoplasia, and a complete absence of the anterior commissure. Sequencing and bioinformatics analysis found a shared region of homozygosity on chromosome 2. Within all large homozygous blocks, very few variants passed our threshold for further analysis, and CTNNA2 was found mutated in all patients. Further, the affected individuals all had very severe homozygous mutations at the C-terminal end of the protein. So we predict that alpha N-Catenin mutations are causative for the patient phenotype. Alpha N-Catenin is encoded by the gene CTNNA2, and there are 3 alpha-catenin paralogs in humans with varying degrees of similarity. The catenin proteins are most similar to vinculin family proteins and share 3 vinculin homology domains. Alpha E-catenin is the most widely expressed and is found within epithelial tissues where it's required for epithelial integrity and polarity. It has also been widely studied. Alpha T-catenin is 55% homologous with alpha N-catenin and is expressed within the heart and the testes. So in human tissue, we found CTNNA2 is predominantly expressed in neurons and at a low level within neural progenitor cells. So similar to what I showed you for known lissencephaly disease gene DCX a couple slides back, the alpha-catenin knockout and spontaneous alpha-catenin mutant mice have been generated and do not display a cortical phenotype by gross histological examination. BRDU birth dating during neurogenesis also showed a normal number of neurons are created and migrate to the proper location within the cerebral cortex. Therefore, we decided we needed to create a human in vitro lissencephaly disease model to understand how patients with these mutations develop the disorder. We generated iPSC clones from 1 affected and 1 unaffected individual from one of our families with CTNNA2 mutations. We also developed iPSCs from a patient with a known mutation that causes Miller-Dieker syndrome as a control. And we also created 3 isogenic alpha-catenin knockout H9 ESL lines that we use also as an additional control. So to assess migration, we modified a traditional neurosphere assay protocol. The way the protocol works is you dissociate adherent NPCs to create neurospheres. We then remove the FGF to induce differentiation. 2 days later, we can place the neurospheres on PLO laminin or matrigel-coated plates or slides, and then 2 days after plating, we can assess the distance that the neuronal cell bodies migrate away from the edge of the neurosphere to quantify neuronal migration. We then performed this assay on control patients, as well as patients with Miller-Dieker syndrome deletion and patients with alpha-catenin variants, as well as our knockout NPCs. Similar to what was recently shown in the 3D organoid model of Miller-Dieker syndrome from Tony Winshaw-Boris's lab, we found roughly 55% of neurons in the Miller-Dieker syndrome patients did not migrate farther than 100 micrometers after a 48-hour period. The alpha-catenin mutant cells showed a similar distribution of migration distances and cellular phenotypes in vitro, suggesting that our model is representative of the patient phenotype of lissencephaly and could be a useful tool to better understand the disorder. We next imaged the neurons during migration and found the alpha-catenin mutant neurons showed very unstable primary neurites and collapsed frequently, impeding their migration. The affected cells also showed a thickened, short neurites with proximal ectopic filopodia and lamellipodia. Their growth cones were wide and heavily branched compared to controls. This phenotype and migration could be restored by reintroducing alpha-catenin expression in the mutant cells using a lentiviral rescue vector. This result showed that these phenotypes were specifically due to the loss of alpha-N-Catenin. So next we wanted to use these cells to study the disease mechanism. The homologue of alpha-N-Catenin, alpha-E-Catenin, has been well studied in epithelial tissues and has been shown to regulate beta-catenin target gene expression, bind beta-catenin and cadherin to control epithelial adhesion, bind and bundle actin filaments, as well as inhibit ARP2/3 polymerization activity. Therefore, we wanted to test whether any of these cellular processes were disrupted in the alpha-catenin mutant cells. We first assessed beta-catenin signaling by performing RNA-seq on NPCs from Miller-Dieker syndrome and alpha-catenin mutant patient cells. We found very few transcriptional differences, which mostly appeared to be due to the genetic background of the patients rather than an effect of alpha-catenin on transcription. To be sure, we also checked Wnt target gene expression by qRT-PCR with and without Wnt3A stimulation and found no significant differences in expression. This suggests that alpha-N-Catenin does not regulate Wnt signaling in NPCs. Alpha-E-Catenin also binds to beta-catenin to control cell adhesion and polarity, so we wanted to test whether loss of alpha-N-Catenin altered neural epithelial structures. So we developed neuronal rosettes from our control and alpha-catenin patient NPCs and assessed the presence of apical markers of polarity. We found normal expression of tight junction marker Z01 and the adherence junction markers of N-cadherin and beta-catenin. In addition, the rosettes showed asymmetrically localized primary cilia at the apical surface. Together, these results suggest that alpha-catenin does not adversely affect neural epithelial polarity or integrity in culture. So the last thing we wanted to check was whether alpha-N-Catenin played a role in actin regulation. We first tested whether alpha-N-Catenin can bind to and bundle F-actin. So we used purified proteins and performed an actin binding and bundling assay. At high speeds, polymerized actin is pelleted, while alpha-N-Catenin is found in the supernatant. But when alpha-N-Catenin is added to F-actin, it binds and comes down in the pellet. Similarly, under a lower speed, F-actin is predominantly found in the supernatant as alpha-N-Catenin. When alpha-N-Catenin is added to F-actin, it binds and bundles F-actin to increase the amount of F-actin in the pellet, which now also contains alpha-N-Catenin. So now that we know that alpha-N-Catenin can bind and bundle actin, we want to determine whether the loss of this function is leading to our cellular phenotype. But first I need to describe how actin is regulated in the cell before going into further detail. So extracellular stimuli control depolymerization of actin filaments by controlling the activity of cofilin, which cleaves F-actin to form G-actin. Monomeric actin is poised for polymerization by binding to profilin and ATP. External stimuli activate WASP, which when bound to ARP2/3 serves as an actin nucleating factor to form branched actin. G-actin can be added to the plus end by formin proteins to elongate actin filaments. Capping proteins can bind also to these plus ends and inhibit formin-mediated actin polymerization. So the process of actin polymerization and depolymerization is called treadmilling. So the majority of molecules that regulate actin polymerization and disassembly are the same in neurons as other cell types, but there are some additional specialized players in neurons. In neurons, actin is predominantly found within the growth cone. Bundled actin filaments create protrusions called filopodia, and branched actin networks create lamellipodia and the actin arcs that are found in the transitional zone at the base of the growth cone, which is necessary to control microtubule advance during neuronal translocation and migration. Based upon the cellular phenotype of the patient migrating neurons, we suspected that actin branching and bundling may be misregulated when alpha-catenin is mutated. But to specifically test whether a direct alpha-N-catenin-actin interaction was necessary for neural migration, we created a couple of lentiviral constructs. The first is alpha-N-catenin lacking the actin binding domain, which is circled in green. The second construct is just the actin binding domain of alpha-N-catenin, which is circled in red. We transduced the NPCs and performed the neurosphere assay in control and alpha-catenin knockout cells to measure migration. The presence of the actin binding domain was necessary to rescue migration. And we also found that the expression of the actin binding domain in alpha-N-catenin was sufficient to rescue migration in the mutant cells, and it also induced migration in the control cells, suggesting that this domain of alpha-N-catenin mediates migration. So since alpha-N-catenin could regulate actin in a variety of ways, we wanted to test whether it was capable of repressing ARP23-mediated actin branching, so we performed an ARP23-mediated actin polymerization assay. Using purified proteins, we can induce actin branching and polymerization by adding ARP23 and its activating protein NWAS, which is shown with the green line. Without ARP23, basal levels of actin polymerization is observed, and this is the black line. But when we add alpha-N-catenin in a dose-dependent manner, we can observe a stepwise reduction in the amount of ARP23-mediated actin polymerization, shown in red lines. So this suggests that alpha-N-catenin can inhibit ARP23-mediated actin branching to regulate actin structure. This occurs without interfering with global F-actin polymerization, since the ratio of monomeric to polymerized actin is unchanged. So this suggests that alpha-N-catenin regulates specifically the ratio of branched and bundled actin filaments in neurons. If alpha-catenin regulates ARP23-mediated actin branching, we wanted to next assess whether our patient cells had increased amounts of the ARP23 complex associated with their actin filaments. So we performed an actin affinity pull-down and measured the levels of ARP3, which is one of the proteins that binds and gets incorporated into the actin filaments during actin branching. We found almost a twofold increase in the amount of ARP3 associated with actin. This suggests that our patient cells have more actin branches or have more ARP23 complex bound to their actin filaments. So if the actin binding domain of alpha-N-catenin mediates this, we should be able to reverse this effect by expressing just the actin binding domain of alpha-N-catenin. So when we performed the same assay in the knockout neurons with and without the actin binding domain, we found expression of the actin binding domain was sufficient to potently block this interaction in vivo. To show this was direct, we also performed an ARP23-mediated polymerization assay and found a dose-dependent decrease in actin branching in the presence of alpha-N-catenin. So our results this far would predict that inhibition of ARP23 would rescue the cellular patient phenotype. So to confirm our model, we identified two different cell-permeable small molecule inhibitors of the ARP23 complex proteins. After a dosing curve, when added to patient neurons for 48 hours, they completely restored neurite length of alpha-catenin mutant, but not the Miller-Dieker syndrome cells. This result suggests that ARP23 overactivity underlies the neuronal phenotype in our patients. So in summary of what I've showed you thus far today, animal models have significant differences in brain development that may make them unsuitable models for certain human disorders and diseases. A viable alternative has been proven to accurately model human brain disorders is the use of human pluripotent stem cells, which have their own distinct advantages and disadvantages. These cells can be used to both identify disease mechanisms as well as test therapeutic candidates to treat these devastating brain disorders. I also showed you an example of 3 families that had a unique brain disorder of pachygyria. We used an exome sequencing approach and find bi-allelic mutations in alpha-N-Catenin. We also used a human stem cell-based disease model to test for neuronal migration defects and also identify the underlying disease mechanisms. We found that alpha-N-Catenin's main function in neural migration was to inhibit ARP2/3-mediated actin branching, without which leads to defective migration. We also showed that loss of alpha-N-Catenin did not affect neural epithelial cell polarity or impact Wnt signaling activity within the neurons. Therefore, in our model, we predict that alpha-N-Catenin normally binds to and bundles F-actin, which controls ARP2/3 branching and the conformation of actin during neuronal migration. When alpha-Catenin is deleted, we have excessive ARP2/3 binding and actin branching occurring. This reduces the stability of the actin cytoskeleton network by preventing bundled actin formation. Therefore, when ARP2/3 is repressed in the alpha-Catenin mutant cells, we can block actin branching, and this allows restoration of actin bundling through other protein interactions and the formation of a stable F-actin network. So lastly, I want to thank all the people that participated in this study, in particular the research participants that shared their clinical data and donated samples that made this work possible, as well as members of my lab who performed the work, and members of the Gleeson lab, which is located at UCSD, who contributed to our findings, the clinical collaborators that have been instrumental in patient recruitment and phenotype interpretation, and our funding sources for this study. And thank you. I'd be happy to take any questions that you might have.
All right, thank you, Ashleigh, very much for that informative presentation. We now begin the live Q&A portion of the webinar. Again, if you have a question that you'd like to ask Ashleigh, please do so now. You can click on the Ask a Question drop-down box located in the far left of your presentation window, and then type the question in, and then click the Send button. Okay, so we do have a couple questions. The first one is about your stem cell models. Did you find any difficulty with any particular starting cell population, like any characteristics it says you may have that may have led to the differences, for example, patient-specific differences?
Well, luckily for this project, alpha-N-Catenin is exclusively expressed in neurons, and so it's not expressed in stem cells. Therefore, the stem cells that we generated from the patients did not have any defects associated with the loss of this protein. We did notice later when we were developing our neuronal cells that there was a bit of variation from line to line. We had to be very careful to make sure that we used neuronal cells that were derived from the same set of differentiations, as well as neuronal cells that were roughly a similar passage number, because they seem to, once they begin to differentiate or they're a little older, we found that removal of the FGF increased, like basically the speed at which they matured varied depending on the passage number of the NPCs that we were using. So we had to be very careful to use representative samples that were processed in parallel, but we didn't notice anything that was specific to the gene mutation itself.
Okay. All right. The next question, if you could have any characteristics about your stem cell model, be it biologically feasible or not, what would it be?
If we could have what? I'm sorry.
Oh, it's okay. It's okay. If you could have any characteristics about your stem cell model, be it biologically feasible or not, what would it be?
I think that, what was great about the model that we really love compared to some other models that might be available is the speed at which you can conduct the research and discover the answers. And we greatly appreciate the ability to take iPS cells and be able to turn them into almost any cell type of the body. There's wonderful differentiation protocols to be able to produce many different types of neurons, as well as supporting cells for the nervous system. And so this has really been a huge asset to neuroscience research in general, and has really increased the speed at which we can discover these disease mechanisms and figure out what's going on in these patient disorders. So I would say probably the speed.
Yeah. Okay. Does your research have any implications into pediatric patients?
Yeah, so I think a lot of these patients, these specific ones that I talked about today, they have very severe mutations in alpha-aN-Catenin, but this gene has also been linked to other neuropsychiatric disorders like intellectual disabilities, schizophrenia, and it's possible that the same disease mechanisms that we discovered with these families are also occurring within these patients with other types of disorders, just maybe in not as severe of manner. So what we like to think of is that we can use these very severe cases of patients with structural brain defects to really learn about biological mechanisms and pathway dysregulation, and that this knowledge can be used to develop therapeutics for patients that have similar disease mechanisms, but maybe not the same exact mutation, but maybe the same pathway is being dysregulated. And so we think that our research would be applicable to a broad set of patients with neurological disorders in general.
Mm-hmm. Okay, that's wonderful news. Okay, just had another question come in. Can patient folding phenotypes be recapitulated well in organoid models?
So there's been some work from Tony Winshaw-Boris's lab, which is also at Case, looking at cerebral organoids, studying Miller-Dieker syndrome, which has the complete loss of folding. I believe some of the mechanisms that they find are most easy to model have been changes in cell number, as well as looking at migration in the organoid itself. They're not really observing folding pattern differences because the organoids are very variable, I believe, in their structure. So it's difficult to have recapitulate the same folding structure over multiple organoids. So you can study the underlying mechanisms of the loss of the folding, but to actually observe the folding happen in an organoid model is, I think, not yet possible.
Okay. Okay, we have another one here. Do you find neurodegeneration in any other mechanism which occurs with as much prevalence as the migration patterns?
Yeah, so
okay.
In this disorder, we don't anticipate that there is neurodegeneration, but we know from the field that neurodegenerative diseases are much more common within the population than lissencephaly. There are some instances of microcephaly with also gyration defects that can be due to a degenerative mechanism or loss of neuronal cells during development or postnatally. But the migration the migration pattern is very specific and also a very rare condition in humans.
Okay. Okay, we've got another one. Do you think, please, I hope I say this correctly, do you think pachygyria and polymicrogyria share common mechanisms?
I believe that they do share a common mechanism of a migration defect. I can't, I have not seen a situation necessarily where patients have the same gene mutated but have a diagnosis of polymicrogyria or pachygyria. So I'm not sure that the same genes are going to be identified, but the clear underlying mechanism in both disorders is impaired neural migration.
Okay. And we have another question. Based on the mouse versus human modeling, could it mean that the mechanisms of neural migration diseases differ between rodents and humans?
That's a really good question. I think we don't know that much yet about migration in humans. Only recently have we been able to really look at human neurons migrating in a dish. There's been some great work out of Arnold Krigstein's lab using viral approaches to look at migration and how they migrate. There's been a couple prevailing hypotheses about why we can make these mice that have these human mutations and they don't really have a phenotype. And one is that that there could be this genetic redundancy, right, which I talked about, and that's still unknown because we know that there's a lot of genes that are human-specific. We also know that mice don't have all the same, or the prevalence of the same cell types within the cortex, and they don't exhibit a folded brain structure. It's also been hypothesized that some of the difference in the brain structure between mouse and humans is due to the distance that the neurons have to migrate. So within a mouse brain, the distance from the ventricular surface to the cortical plate is much smaller than what you would see in a human brain. And so that the distance really makes up why the phenotype is observed in humans and not in mice. But this hasn't really been tested. I think recently people are also starting to, put human neurons or progenitor cells into mouse brains and look for changes in migration. But again, we don't know if all the cues that direct migrating cells are present in mice and are expressed at the same level or present in the same place as what they are within the human brain. So I think this is an area that's going to have a lot more focus in the future to really understand why we don't really have any good animal models for a lot of these types of human brain disorders.
Okay, and actually just a question from me on that, and just thinking about the assays you need to do to measure migration. Is it very difficult to do that in mouse cell assays in the way that you've done them for human cells?
Actually, I think they were done originally in mouse cells, and that's where the assays originated from. And in that case, what you do is you take the isolated mouse progenitor cells and you form neurospheres, and you remove the FGF, but you don't really have to age the cells. They mature at a different rate than the human cells. So when you directly plate the neural progenitor cells, they can migrate out of the neurospheres, and you can see accurate changes in migration. But what's still unknown is if you would take these NPCs from a mouse that has a mutation in a lissencephaly gene, and if you did the same exact assay with the human cells and the mouse cells, would they act the same, or would the human cells act differently than the mouse cells? So there hasn't really been anyone that has investigated that phenomenon yet. So I think there's still like a lot of assays to be developed. There's also different types of migration in neurons. There's neurons tend to migrate along the processes of radial glial cells. And so our assays are artificial in the fact that, you know, you're plating neurons on a matrigel plate and they're not migrating along these fibers. So there's also people interested in looking at migration along these fibers as a different mechanism of migration than what might be observed within these culture systems.
Okay. So I do have one more question here, but I just want to open it up again to the listeners. If you've got a question, please type it in. We do have some time. And Ashleigh, the next question was, can you tell us a little bit about your next steps for research? If you're, yes. If you tell us a little bit about your next steps for your research.
Sure. Our lab is really focused on trying to identify new types of pediatric brain diseases and trying to identify what those mechanisms are that underlie the disorders and whether there's any commonalities between these different clinical syndromes and spectrums. And so we are focused on recruiting additional patients that might have these pediatric brain disorders of unknown genetic etiology, where we can help them identify the molecular cause, as well as pinpoint like therapeutics that might be helpful for them in the clinic. And then we like to combine using these cell-based models with animal models, and also our tissue culture models with using iPS cells and their derivatives, their neuronal derivatives. So a lot of our diseases that we study are actually quite well modeled in mice, and so we also try to validate our approaches using animal models whenever possible. So we're trying to identify new mechanisms of neurodegeneration in children, and we're trying to expand upon some of our previous findings and learn about whether some of these disease mechanisms are actually common to more prevalent disorders like autism or intellectual disability or schizophrenia.
All right. Sounds very exciting. And there are no more questions at this time. So once again, I would like to thank you, Ashleigh, for your presentation. It was great. We would love to have you back. It was great to hear you.
Thank you.
And before we go, please note, everyone, that today's webinar will be available for on-demand viewing through February of next year. And thank you all for joining us. We hope you enjoy the rest of today's event.
Thank you.